Good morning, everyone, and welcome to the third talk of the cycle of conferences entitled Chemistry, a Hard Outreach to Society. Our speaker today is Begoña Lavín Plaza from the Biochemistry and Molecular Biology Department, and the title of the talk is Journey Inside the Body, How to Study Cardiovascular Diseases Using Magnetic Resonance Imaging. We have chosen the online format instead of face-to-face, taking advantage of the platform provided by the General Foundation of the UCM, to whom I want to thank the help and support to organize this activity. In addition, we thank to our funding sources, the Spanish Biochemistry and Molecular Biology Society, the Royal Chemistry Society, Sectorial of Madrid, and Vice Rector of Research and Transfer for their financial support. This enables us to use this format, and this way many people will be connected. Our junior researchers who have been investigating different aspects of the chemistry have returned to our institution and spending a long period abroad. This is one of the reasons we organize these webinars in English. Therefore, we are able to broaden our audience to foreign institutions, some of which have been the host institutions for these researchers. The lectures take place on Thursday, every Thursday, at 12:30, and will be announced every week, including the link of the registration form. Today, I will include the link of the fourth talk of October 14 in the chat. Students who have enrolled for credit for this activity should email me and the address given in the announcement of this activity. Without further ado, I give the turn to the moderator of this talk, Juan Manuel García Segura, Professor of the Department of Biochemistry and Molecular Biology, who will introduce the speaker and moderate the questions that you can write in the Preguntas and Respuestas button. Professor García Segura, it's your turn.
Okay, thank you. Hello, everyone, and thanks for being there online. It is my pleasure to moderate the talk of today with Dr. Begoña Lavín as a speaker. Begoña joined our Department of Biochemistry and Molecular Biology at the beginning of this year, and after this relatively short period of joint work and coexistence, I can certainly say that we have been lucky and successful with this incorporation. Something that I think you will be able to see for yourself as you get to know Begoña. So now I'm going to introduce her to you by telling you the highlights of her training and career, and then she will tell us about her lines of work and her know-how. Begoña Lavín Plaza obtained her bachelor's degree in biology in 2008 and her master's degree on therapeutic cell targets in 2010, both from the University of Alcalá de Henares. Between 2008 and 2013, she completed her PhD at the Centro Nacional de Investigaciones Cardiovasculares, our CENIC in Madrid, with research focused on the effect of proteolytic enzymes and nitric oxide on vascular remodeling. During her PhD, she completed an internship in Professor Bodnar's laboratory at the School of Biomedical Engineering and Imaging Sciences at King's College, London in 2012. After finishing her PhD in Madrid, she returned to this laboratory in London, where she has carried out a long postdoctoral stay from 2013 to the beginning of the present year, 2021. Her research interest is focused on deepening the knowledge of the molecular mechanisms underlying the role of vascular inflammation in different cardiovascular diseases, as well as identifying new biomarkers that are useful as therapeutic agents or imaging probes to treat or diagnose cardiovascular diseases. To date, she has published 21 high-impact articles, 2 book chapters, and 1 international patent. Likewise, She has presented numerous abstracts at national and international conferences, including 10 oral presentations. In December past year, 2020, she obtained in a public competition on merits the position of Profesora Ayudante Doctor that she currently holds in our Department of Biochemistry in this Faculty of Chemistry, where she is currently establishing a new area of research focused on the interaction between cardiovascular disease and inflammation. Okay, so far what I wanted to tell you about Begoña Lavín. Now, Bego, it's your turn, but first, let me tell you many thanks for your presence here today, and also that we are eager to hear your proposal to travel inside the body using magnetic resonance imaging to study cardiovascular diseases. So, Bego, whenever you want.
Thank you very much, Juanma, for the very kind introduction. And also thank you very much for the organizers of this series of seminars, and especially to Maite, who invited me to give this presentation, which I'm very, very happy to do it here today. So let me share my screen. So I hope you can see well the presentation and also hear me. So yeah, today basically I'm very, very happy to show you all some of the results and some of the work that I was doing as a part of my postdoctoral training in London during the last 8 years. And as I will show you today, not COVID-19 is not the only pandemic we have in our society. Cardiovascular diseases actually kills many people every day, but also leave a lot of people with important disabilities. However, I will also show you that we have a strong methods for the clinicians to be able to detect and also treat these cardiovascular diseases in a way that is going to be non-invasive. But obviously, as all research, we need to put more efforts in developing these techniques. So at some point, we will be able to have stronger, very, very strong methodologies that will help clinicians to take care of the patients. So to start with, I will show you some statistics so you will can understand why it's so important to study cardiovascular diseases. So in this first graph, you can see the leading causes of death globally, worldwide. And you can see that the number one is ischemic heart diseases. And the second one is stroke. Both of them are cardiovascular diseases. If we take a look about what happened in our country, what happened in Spain, here are the top 10 causes of death. And we have ischemic heart diseases, again, in the first line. And we have stroke in number 3. And we also have hypertensive heart disease as number 9. So 3 out of the 10 causes of death in our country, or leading causes of death in our country, are cardiovascular diseases. But let me show you some striking statistics. Taking as an example, United Kingdom, who does quite a lot of statistics, and they check in very little detail everything. So I will tell you that in the United Kingdom, around 80% of the people that have cardiovascular diseases, they also have another health condition. So you can imagine how much problem these patients can have to have normal lives. Today, in United Kingdom, 420 people will die because of cardiovascular diseases. And from this number, 110 will be younger than 75-year-old. Also, there are 7 million people living actually with cardiovascular diseases. But if we want to be a bit more strong with the data, I can tell you that today 540 people will go to the hospital because they are having a myocardial infarction. And from this number, 180 will die. And a very sad statistic is that also 12 babies will die because of will be diagnosed of heart defects. All of this means that we will have 1 death in the United Kingdom every 3 minutes. And I can tell you that in Spain is not much different. But what are cardiovascular diseases? And specifically, what is atherosclerosis? Atherosclerosis is known as a silent disease. And it's because we are not going to feel anything until the situation is very severe. If we want to define in a very easy way what is atherosclerosis, we can say that it's a buildup of fatty material inside our arteries. And this is basically what I'm trying to represent in this diagram. It's important to tell you that we develop atherosclerosis from our childhood until we die. It's a very long process. And these plaques, this accumulation of fatty material is generally known as atheroma or atherosclerotic plaque or just atherosclerosis. So what happens when we have a plaque or atheroma in one of our arteries? First thing, and I think you can see it from the diagram, is going to be an arterial narrowing. So meaning that the blood is going to have less space to go through. But this is also going to cause what is called arterial hardening. If you think about an artery is quite elastic and it's going because it needs to do vasodilation and vasoconstriction. And when we have a plaque, this vasodilation and vasoconstriction is affected. Also, as we will see in a bit in the next slides, it can happen when a plaque breaks, it can have a thrombus formation. And this thrombus formation can cause a complete blockage of the artery. So nutrients and oxygen are not going to be able to arrive to the tissue that they need to arrive. And if this happens in an artery in the brain, it's going to cause a stroke. And when the blockage is done in the arteries that are in the heart, is a myocardial infarction. So let me show you in a very short video what happened in a myocardial infarction situation. So here we have our heart, and we can see here the coronary arteries that feed with oxygen and nutrients the tissue. In this coronary artery, there is a plaque. So there is less space for the blood flow to go through. And imagine that this plaque breaks. So it's going to be a clot formed by platelets. And also at some point it's going to completely obstruct the blood flow. What happens under this blockage is that the tissue that is underneath is going to die. All the myocytes are going to die. And this is a myocardial infarction. And this will never be recovered. It's something that dies and it will never come back, or is not going to be replaced by new cells or anything. But I was telling you that we are producing plaques all of our lives. And you might ask me, but Bego, are all our plaques going to break at some point? Well, thankfully not. And this is what I'm going to try to explain you here. We have different types of atheroma plaques. The ones that are known as stable, which generally are not going to break, and those ones that are vulnerable, and these ones are the ones that are prone to rupture and cause severe problems. So as you can see just by the diagrams, they look quite different, and we are going to see in details these differences. So first one, we have the extracellular matrix components that we have in both plaques. In general, is something that we can think is going to provide potentially stability on the plaque. We also have something called the fibrous cap that we can see that is like a layer or a barrier that is going to separate the fatty component from the blood. And we can see the most important thing is that we can see that the fibrous cap in the stable plaques is quite thick. However, in the vulnerable plaque is quite thin. We also have inflammation inside the plaques. And as you can see, inflammation is much higher in vulnerable plaques than in stable plaques. Another component is the lipid core, which is basically dead material. And generally, the lipid core is much bigger in the vulnerable plaques than in the stable plaques. Another component is the calcification. And actually, in stable plaques, we usually find macrocalcifications, which provides with quite a lot of stability. However, in vulnerable plaques, we find what is known as microcalcifications, which basically can make ruptures within the plaque, making them more vulnerable. But we also have three components that we find in vulnerable plaques, but not in stable plaques. The first one is the thrombi formation. When this fibrous cap in the vulnerable plaques, that remember is thinner, it breaks, a platelet deposition is going to kind of try to cover this little injury that has been created, and it can create a thrombi that potentially can completely blockade the artery. We also have some internal hemorrhages in the plaque, which is also going to provide instability. And finally, we also have what is known as neovessel formation, new little vessels that are going to be created within the plaque that are going to feed the plaque with more nutrients and oxygen, so the plaque will be able to grow faster. But what is the role of imaging? At the end, the topic of today is to use imaging to study cardiovascular diseases. So what is the role of imaging on cardiovascular diseases, to study cardiovascular diseases? So here you have like a diagram of the progression of an atherosclerosis. And depending if we are in early atherosclerosis, in vulnerable atheroma, or already in myocardial infarction, we can visualize different biological changes. For example, in early atherosclerosis, we can check how big our plaque is, or how permeable is for the inflammatory component to extravasate into the vessel wall, or we can check inflammation itself. When we are in vulnerable atheroma, we can check for the luminal area. So the space that we have for the blood to go through, that is going to be an indirect measurement of how big the plaque is. We can also check again for inflammation, or if the plaque already broke, we can check for thrombi formation. And when we are already in myocardial infarction, for example, or a stroke, we can check for the neovessel formation, for fibrosis, or for thrombosis. So we have different non-invasive modalities to do this imaging. But today I'm going to try to convince you that magnetic resonance imaging is one of the best methods that we can use for this kind of diseases. Why? Because first of all, it's non-invasive. The second one is because it doesn't require radiation, like for example, PET. We can also have very high resolution images, and we can do tissue characterization without or with contrast agent. And more specifically today, we are going to focus on the development of molecular MR-specific contrast agents. So what are the ingredients of cardiovascular imaging? So to be able to develop a new contrast agent, we need to follow some steps. So first of all, we need to decide what is our molecule of interest, which molecule is going to provide us information that will tell us in which stage of progression the disease is. So this we do it with the target screening. When we choose a molecule of interest, then we will move to the chemistry to try to design a contrast agent that is going to be able to bind to our molecule of interest. We will validate with chemistry this contrast agent, and then when we are happy with that, we will move to the biology and animal models of preclinical imaging, where we will reproduce in animals different cardiovascular diseases in order to test how our contrast agent is working. After that, and once everything is kind of approved, we move to the clinical stage, where it's very important the imaging technology to improve all the materials and all the machines that we use. It's very important the data processing to be able to get the maximum out of the images that we obtain and the clearer as possible. It's also very important to, and it's now very trendy to do machine learning. Basically, machine learning is in the stage of trying to speed up the process of imaging and diagnosis. And finally, that will lead to improved patient care. Today, in this talk, we are going to focus in this first part. And at the very end, I will try to convince you why it's important to use contrast agents, showing you a couple of examples of patients. So if we move to the first step, which is the target screening. Target screening can be done in many different ways. We can use either proteomics, genomics, metabolomics, histology, or actually we can go to the literature, what is published there, and check which molecules could be of interest to us. So here I'm showing you a cross-sectional section of an artery where we can see in black 3 lines which represent mature elastin. Remember that arteries are very elastic, as I told you before, and it's because they have elastin, so allows vasoconstriction and vasodilation. So we can say that we have quite a lot of elastin in a vessel wall, and it's highly abundant enough to be able to detect it with magnetic resonance imaging. But let me show you what happens when we have a plaque. When we have a plaque, we can also see that there are new fibers more or less everywhere in the plaque. So there is a net increase of elastin in atherosclerotic plaques. So let's see a bit the biology or the elastin turnover of in cardiovascular diseases. So here we have a control tissue. And in control tissues, in normal healthy tissues, we have all of our elastin mature and cross-link. In a diseased tissue, what can happen first is that there is a degradation of this elastin into little peptides. For example, this can be done by proteases or elastases, creating some elastin-derived peptides. But also in disease happen a second thing, which is the novosynthesis. So the precursor of elastin is known as tropoelastin and are little monomers. These monomers, in normal conditions, they are going to be cross-linked to be like in the control tissue. And this cross-link process is done by an enzyme called lysyl oxidase. However, in disease, lysyl oxidase is downregulated and tropoelastin is accumulated. Let me show you that in this graph. In normal tissue, we have very little tropoelastin. In plaques or aneurysms, we have a net increase of tropoelastin monomers. Similarly, in normal conditions, we have our LOXA mRNA levels high, and when we have the disease, this is downregulated. Let me show you another example from our experiments. Here we have an example, an histological example of our aorta from a rabbit, and we can see in black quite a lot of elastin fibers. When we check for tropoelastin in the same segment that we should see it in brown, we don't see much because it's a healthy vessel. And here we can see the segmented tropoelastin, there is almost nothing. However, when we go to a diseased artery that has a plaque, we still can see some elastin, but we can see in brown quite a lot of tropoelastin, also segmented here, in the plaque. So there is a net accumulation of tropoelastin. What I'm trying to tell you is tropoelastin might be a very cool target for magnetic resonance imaging. So then, after deciding that tropoelastin was our molecule of interest, we moved to the chemistry. And we developed 2 potential candidates. So the first candidate, we call it BBGS, and is formed, here we have the structure of the contrast agent, is formed by the amino acid sequence that binds to tropoelastin from the elastin binding peptide protein. So here basically we have our sequence of amino acids, and then we have here a DOTA, which is, for those that are not familiarized, is like a box. And inside the box, we put the gadolinium. The gadolinium is what is going to give us the signal in the MRI. Gadolinium cannot be free because it's very toxic. So we keep it in a little box. So our second candidate was the one that we call YPDH. And it came from the interaction of tropoelastin with the MACP1 protein. Similarly, we have here the sequence of amino acid that binds to the tropoelastin, and then again we have our DOTA with the gadolinium. So basically we have two potential contrast agents, BBGS and YPDH. Now we need to test, for example, for affinity, which one binds better to tropoelastin. And for that, we calculate the KD, that is the constant of affinity. So first, we did different binding experiments, and we can see that the YPDH contrast agent binds to tropoelastin and also to mature elastin. And yeah, it's different, but what we saw is that when we were using the BBGS, the discrimination between tropoelastin and elastin was much bigger. And also the KD, which means the affinity to the contrast agent to bind to tropoelastin, and the lower the KD, the higher the affinity, we could see that BBGS is better or has a better property of affinity, of better affinity than the YPDH contrast agent. We also did one experiment with the BBGS contrast agent where we replaced the gadolinium DOTA and we put a rhodamine. And what we did, it was an immunohistochemistry to see in this aneurysm where tropoelastin was, and we could see that it correlates quite nicely with the contrast agent label with rhodamine. And we could see it in the disease, but not in the control. That, remember, we don't have in healthy tissues tropoelastin. But okay, now we know that both our contrast agents are binding to tropoelastin, but it's very important to know how long it takes to clear the contrast agent from the body, from blood. And, you know, we need to have a contrast agent that stays in the body enough time to be able to do the imaging, but we don't want this contrast agent to be there forever. This needs to be clear kind of quick. So the first thing we did, it was to study the blood clearance. And we could see that both contrast agents behave quite similarly. And after 1 hour, the clearance was almost complete. In these PET images, here we have BBGS labeled with Copper64, and here the second candidate, YPDH. We can see the full body of the mouse, and we can see the kidneys, and the bladder. So it's telling us that the contrast agent we are getting rid of the contrast agent through renal excretion. However, we could see that in the YPDH, some of the contrast agent got stuck in the liver, which we are not interested in that. And in the BBGS case, it was quite clear. So taking this into account, we thought, okay, probably our best candidate is the BBGS contrast agent, that from now onwards, we are going to call it gadolinium tesma. So with this gadolinium tesma, we move to the preclinical or animal studies. So before we go into the animal study, let me show you how a scan with a mouse looks like. So here we have our little mouse that we are going to inject the gadolinium tesma contrast agent. And then we will put it in the scanner. This is a clinical scanner. Patients are scanning this scanner every day. And the reason why we put our animals in is because scanning in a clinical scanner is going to help in a quicker translation of our contrast agent. And you might ask me, but Bego, where is the animal? Well, the animal is there, but we need to zoom in a bit. So if we zoom in here, we can see many different things. So the first thing we see is a coil, which is an instrument that the MRI has that is going to receive the signals and is going to send the images into the computer for us to see inside the animal. We also have the Ecg Module that you can see it better in here, is here. And this one is going to trigger or record the heart rate of the mouse. A mouse has a heart rate between 400 to 600 beats per minute, quite fast. We also have a mask that is going to provide anesthetic, isoflurane in this case, to the animal. So all the scanning session or the imaging session, the animal will be asleep and not suffering, of course. And then we have also the water tube, this water tube that is going to have warm water that is going to help to keep the body temperature of the animal at 37 degrees. And then if you see this black dot over there and maybe a little tail over here, there is the mouse. So we take the animal, we put it in, and we start our imaging session. So let me show you now how we do the scans of the vessel wall using MRI. First thing I would like to tell you is that it takes, if everything goes well, it should take around 50 minutes. So first, we sleep the animal, we inject the contrast agent, and we put him in the MRI. First images that we are going to take are called scouts. We take one coronal and one transverse. In the coronal, we can see here the head. For example, here we can see a bit the heart and the liver are not very clear images. We just want to more or less see where is our region of interest. In the transverse section, we can see 3 bright dots here, which are the brachiocephalic artery, the left carotid, and the subclavian. After we are happy and we say, okay, we are going to scan in this region, we are going to run what is called a magnetic resonance angiography. And in this case, with this sequence, we are going to be able to see the spins that are moving, the tissues that are moving. We are going to see them bright, and the only tissue that is moving is the blood. And all of the rest of the tissues that are not moving, are stationary, they will be black. So this allows us to do a very nice 3D reconstruction or delineation of the arteries. So here we can see clearly the aortic root, the arch, the brachiocephalic artery, and the 2 carotids, and 1 subclavian. After this, we run a sequence that is called log locker. And the log locker is going to allow me to detect at what time the signal from the blood is 0. So we will see the blood completely black. We use this time to apply it in another sequence that is called the late gadolinium enhancement. So as you can see, we will see with this sequence where the contrast agent is extravasated into the vessel wall, and we will see the blood black. We can also do nice 3D reconstruction images, and we will be able to visualize where the plaques are, because the contrast agent is going to extravasate only where we have plaques. And finally, we have a last sequence that we use quite often that is called T1 mapping. I'm not going to enter into details, but this sequence is going to help us to characterize the molecules that we have in the plaque. So after saying that, let me explain you that we did the preclinical validation of this contrast agent using 3 different animal models. The first animal model was a mouse, a very known murine model of atherosclerosis progression. Basically, these animals develop in a very reproducible way atherosclerosis in the brachiocephalic artery. So we could study progression of the disease. Then we moved to a bigger animal, to the rabbit model, to study, to be able to see if our contrast agent was able to differentiate between stable and vulnerable plaques. And finally, we did a third set of experiments where we study aortic aneurysms, which is another silent disease that creates like a dilatation on the arteries. So we also wanted to see if we could detect with our gadolinium tesma this disease in mice. So let's move to the first one. Here we have our atherosclerosis model, so we can study the progression of atherosclerosis. We take our mice, we put them in a high cholesterol diet. Basically, we feed them with burgers every day, and we study at different time points. So we have before the burgers, and then at 4, 8, and 12 weeks after high-fat diet. So we can see that progressively there is an increase of contrast agent over time, and this correlates quite nicely with an increase of tropoelastin signal, and obviously with a bigger plaque. We also wanted to test a treatment for atherosclerosis, which is statins. You might hear about them. And we've fed our animals for 12 weeks together with the statin treatment, and we could see that the signal, it was much lower, and also the plaque was much smaller. And this is what is also represented here. Tropoelastin increase over time with high-fat diet, and when we give the treatment, it decreases. So we can say gadolinium tesma can evaluate, or we can study the progression of atherosclerosis using gadolinium tesma. Then we move to the rabbit model, and as you can see here, after injection of the contrast agent, we could see the vessel wall of the aorta. And in this second example, we could see it even a bit brighter, but let's see what does it mean later. We could see it here. I'm more than happy to explain you maybe during the questions how we do this model, all of these models. If you are interested, just ask. But we can induce the break of the vulnerable plaques in this model. And when we do that, that is called triggering, we can see that some regions, they are going to be, after triggering, they look like before, nothing changed. However, we will have plaques that they look like they have a bite in the lumen. And what it has happened is a thrombi formation. Remember, the plaque broke, the platelets go there, they create a thrombi that might be completely occluding or partially, like in this case. And this is a characteristic of the vulnerable plaques. So retrospectively, we can evaluate if these plaques or these plaques, this amount of tropoelastin in these plaques is representative of vulnerable or stable plaques. And what we could see is that, yeah, we could differentiate with our gadolinium tesma contrast agent vulnerable plaques from stable plaques, which we were very happy about that. And finally, we move to the aneurysm model. And here I would like to show you how an aneurysm look like. So we have, this is an aorta of a mouse, and here we have a normal segment, and then we have a dilatation. This is basically an aneurysm. And the problem is that you don't feel anything until this aneurysm breaks. And when it breaks, there is a very, very, very high percentage of people that die because of that. So we wanted to check if our contrast agent was detecting these aneurysms. So again, we did the same, and we could see that in control regions or healthy or non-dilated regions, there was not much contrast agent. However, when we were in the dilatation area, we could see a higher accumulation of tropoelastin. So basically, we could see that gadolinium tesma, it was confined to the dilated areas of the artery. So just to finish with this seminar, let me show you why, what is the clinical importance of MRI using molecular specific contrast agents to improve the patient care. So the contrast agent that I have shown you until now, gadolinium tesma, is in the last stages of the preclinical validation and is going to start the clinical phase very, very soon, which I'm very looking forward to see the results. But I'm going to show you two examples using another contrast agent that is already clinically available and binds to albumin. So here we have a patient that came to the hospital with severe symptoms. He was having basically a stroke. Here we have our scout. We can see the head, the neck, the ears, and then we have the carotids. If I focus on this region and I do a transverse cut, we can see something like that with our magnetic resonance angiography. We can see the outer carotid artery and the inner carotid artery. And when we run another sequence, no contrast agent, we could detect, because of the properties of thrombi, thrombi are bright, we could see that there was a thrombi in the inner carotid artery. But when we injected the contrast agent, yeah, fine, we could see the thrombi, but we could also see that there was a vulnerable plaque in here with an internal hemorrhage. And also, we can do 3D reconstructions to see what is the size and the organization, let's say, of the plaque within the vessel. This kind of information, this increase of information, it can help quite a lot the clinicians to decide what to do or what is the best way to proceed. In this case, the patient came with mild symptoms. He was a bit dizzy, but not much more. So here we can see his carotid. And if I do again the transverse cut, we can see the right carotid artery and the left carotid artery. Again, we could see without contrast agent that in the right carotid artery, we had a thrombi. But when we injected the contrast agent, we could see that, yeah, fine, we have here our thrombi, but it also had a vulnerable plaque that needed to be treated. And also, if you see here, where is the left carotid artery, we cannot see much. However, when we use the contrast agent, we could also detect an unstable plaque that might not need treatment now, but needs to be monitored just in case it grows so much, it's going to create symptoms. And also, like before, we can do 3D reconstructions that will help clinicians to decide what is the best way to proceed with these patients. So to finish, I will obviously, this is not a work that I did by myself, and I want to show you something that I think or is the most important thing that I learned during my stay in London. First is that this work needs to be done between engineers, biologists, chemists, medical doctors. Otherwise, it's not going to work. I'm trying to tell you that it's a multidisciplinary work, and also it's a multi-country work we had because we were people from the UK, from France, from Italy, and also from Chile. And obviously, without our funders, as you can imagine, this work is quite expensive and it wouldn't be possible. So yeah, with that, I hope I convince you and that MRI is a very cool technique. It's very important to study cardiovascular diseases. And with that, just thank you for your attention, and I'm very happy to take questions.
Okay, thank you very much, Bego, for this nice presentation, for this nice talk. I have really enjoyed it a lot. It's a very nice example of a multidisciplinary approach to study a very relevant, in clinical terms, a very relevant disease, and also in social and population terms. Okay, let's say that it obeys very, very well to the goal of this webinar, because you have put in your presentation a very nice example of how the chemist can outstretch a hand to the society to solve a clinic problem in this case. Okay, I don't know if there are questions already posed by the attendees. Let me check. Okay, I'm going to translate that. Abrahamian asked if you think it's possible to use another element which would be less toxic, and hopefully, he says, cheaper than gadolinium?
Well, it's a very good question, actually. Well, gadolinium itself is not so expensive. What is expensive is the process of creating all the contrast agent and all the validations, and even more when you jump into the clinical phase. Actually, we use in the clinics also not only gadolinium. We can use usually iron particles that are very helpful to target, for example, for inflammation. And nowadays there are also some experiments, let's say. There are not a lot of literature about that, but some groups are starting to use fluorine-19, which is not toxic at all. And because we don't have fluorine-19 in our body, it's a very easy way to image, and it's not going to give us background noise. So yeah, we can use different things, but gadolinium works quite well. And once it's in the box, is within the DOTA chelate, there is no problem in principle.
Okay, thank you. We have another question. I think it's a bit related with the previous one, but in a more general term, because Laura Nogues asked, apart from acknowledging your cool presentation, ask you if could MRI be used in other pathologies or medical conditions?
Well, thank you, Laura. Oh, yeah, of course. I mean, today I show you what I know or what I have been doing, right? That is the cardiovascular diseases. I was actually also doing liver disease, for example, to evaluate fatty liver disease is quite helpful. And obviously, if you think about it, I mean, cancer is a very hot topic. And some of the contrast agent, for example, the tropolastin contrast agent that I show you today, but also the albumin one that you saw in the patients, it can be applied, and we had applied in few models of cancer. So yeah, I mean, I think you have diseases that are more helpful to be used. It can be easier to be used with MRI, like for example, also the brain. I think most of the huge amount of research is done with MRI in brain. But yeah, you can move wherever you want with MRI, more or less.
Yeah, of course. Yes, thanks. Okay, you have, I don't know if there will be additional question, but in the meantime.
I have a question. The problem is that the panelists cannot ask questions by the channel of preguntas and respuestas. Then I have to ask my questions.
Okay, Maite, go ahead.
Okay, thank you. I have two questions. First, I saw that when you showed the affinity of the contrast, I saw that that also the contrast has affinity for collagen or fibronectin, and also even more in the VVGS than the YPDH. Then this can be a problem for the specificity of the contrast.
Well, yeah, it's true. There is also, I mean, we wanted to differentiate mainly between tropolastin and elastin, material elastin. It's true that the contrast agent binds to collagen and fibronectin, but we have also seen that these two molecules increase with the progression of the disease. So we can consider it as an additive. It's true that it's not going to be a super clean measurement of tropoelastin. It's kind of contaminated with some collagen or fibronectin that can bind. But overall, that the goal is to be able to detect or evaluate the progression. It does it quite nicely.
Okay, ready?
I have another. Sorry. I saw that, I mean, you use this technique to follow the atherosclerosis also, but I think that the aneurysm is also a very important disease, that it's even more silent than the atherosclerosis, or even as silent as atherosclerosis. Then do you think that it's a technique that in the near future is going to be a diagnosis tool for this kind of pathologies?
Well, it will be great if that happens. It's true that it's going to jump now into the first clinical phase. But what we saw is that we are able to detect aneurysms, but also we can kind of, based on how much tropoelastin we find in our measurements, we can kind of predict which ones are going to break in the, let's say, short term, what the animal study allowed us to evaluate, which was basically 4 weeks. So it could. I mean, obviously, it's not going to be there in the market tomorrow, because usually it takes quite a lot of time and validation. But it seems that tropoelastin could be a very nice candidate for, and there is nothing out of there to check for aneurysm, so it can be very helpful, we think.
Well, thank you very much. I see that there are more questions, but later if I can do another one. Okay.
Okay, thank you, Maite. We have an additional question, I think, related with the previous one of Maite. Question again from Bagarat Abramian, that he asked, can you use two different contrast agents to clear the image about the protoelastin collagen binding?
Well, it's a very nice question, actually. We can and we cannot. Let me explain you why. So for example, if we use fluorine contrast agents and gadolinium contrast agent, we could do simultaneous imaging if we have the appropriate coil. In the case of, for example, as you asked, tropoelastin and collagen, I can tell you that in the lab, there is now a PhD student, Nadia, who is developing a collagen contrast agent, collagen 1 and collagen 3. And she is gadolinium based. So what we can do in that case is maybe today scan with tropoelastin, wait for the complete washout that we know that is around 2 hours. And then maybe the day after, just to be completely sure, inject with the collagen if it's more specific, and then do a subtraction of the values or the measurements that we get from one or the other. But yeah, we can do simultaneous imaging in some cases. As I said, we have done it with fluorine-19 nanoparticles to measure inflammation. And at the same time, we were measuring matrix remodeling with an elastin binding contrast agent that it was gadolinium based. So yeah.
Okay, thank you. Another question about the gadolinium is posed by Javier García Virseda. I read his message, his chat. He says, really good presentation, Begoña. Thank you so much. Why did you start to study with gadolinium and didn't start with another element, with gadolinium in particular?
Well, we started
Why gadolinium
with,
in particular?
Okay, so again, it's a good question. We started with gadolinium because we use, I mean, I didn't go into the detail, but we have the T1 mapping sequence that is the one that we generally use, and we had quite well defined in the lab to evaluate a gadolinium contrast agent, because it's going to make changes in the T1 mapping sequence. It's true that we could have moved to iron particles, that they are going to make a more strong effect in T2 mapping. But I think most of the contrast agents that are now in development, they use gadolinium because we can see the signal bright. However, when we use iron particles, we see voids. So we see darker. And darker is usually not that nice, because sometimes you don't know if it's that there was no signal or what. So usually gadolinium is what is used in general. Also, we had the lack of having 2 chemists, well, actually 4 chemists that they were very knowledgeable in gadolinium-based contrast agent, who were Sara, Maggie, Federico, and Giuseppe, who they were developing, and they were the contrast agent, and they had all the knowledge there. So that's why we moved there.
Okay, thanks. Okay, I have two questions, methodological questions that I would like to ask you, because you have presented two kind of studies. One, preclinical images, basically on rabbit, clinical images with patients. Okay, about the rabbit, the rabbit model. How do you get some atheroma plaques to rupture?
So the rabbit model is a very established model. So basically we take the rabbits, we do a surgery on their arteries, where we basically enter in from the leg, we go into the abdominal aorta, and we scrape the inner part of the abdominal aorta, and we remove the endothelium. So then we close the animal recovers perfectly. And then we feed them for some time with high-fat diet. By removing the endothelium, we create the maximum permeability. So both fat and inflammatory cells are going to fit inside the vessel wall of that regions. So with that, plaques are going to start to grow. So after some time, what we do is the scanning. That is what you saw in the first part of the slide. And then we need to trigger the rupture of these plaques, because some of these plaques will be stable and others will be vulnerable, but we need to know who is who. So to do that, what we do is an injection of a cocktail that contains a Russell Viper venom that is going to create a kind of, let me say, a kind of a vasoconstriction. And also we are going to inject histamine that, well, the Russell Viper venom mainly creates, is a procoagulant, and also the histamine is going to create vasoconstriction that it happens only in rabbits. It's a vasoconstrictor only in rabbits. In the rest of the animal, it's a vasodilator. So in such way, we create like spasms in the artery, and those ones that are vulnerable are going to break. So that way we can differentiate one to the other.
Okay, thank you. Interesting. Did you do that experiments or? So
the surgeries were done by one medical doctor, and then all the rest it was done by mainly Alkistis and myself and Sarah.
Okay, yeah. Thank you. My other question was about patients. So when a patient arrives at the emergency room, how do you come to know or decide which part of the body or artery needs to be scanned?
Well, that is a nice question, because obviously with MRI, we cannot scan the whole body and then say, okay, let's see where the broken piece is. So what happens generally is the patient arrives to the hospital and he has some symptoms that is going to tell you maybe where the problem might be. Usually another techniques, faster and cheaper, are done first, just to check if it's a myocardial infarction, a stroke, or anything else. And then you kind of localize where the problem might be. So after that is when you can say, okay, it's going to be around the neck, might be the carotids, I don't know, one or the other. So you scan both, but you are already in a region of the body.
Okay, I see. Yeah. Okay, thank you. And my last question, I don't know if there is another one by the attendees, no? Okay, my last question is, okay, you have talked us about contrast agents that are in a preclinical phase, okay? But there are also contrast agents in application, in the clinical application. So what type of contrast agents are there? Which are clinically approved and which do you know are still are still in the preclinical phases?
So yeah, I mean, we have not so many contrast agents in the clinical. We have few, like the one I saw you with the patients with these PROHANs that it binds to serum albumin. So it's an indirect measurement of vascular permeability. We have also some iron particles like ferumoxytol that can also target for immune cells. They use the phagocytosis of the cells. So the contrast agent basically is going to eat them by macrophages and is going to help. So the macrophage is going to get stained with the iron and is going to allow us to see it. These are more or less, so we have few gadolinium, we have then magnetist and other contrast agents that are not so specific. And then we also have many more contrast agents on the preclinical phase. Like, for example, we have this tropoelastin that is about to jump in preclinical phase, in the first clinical phase. We also have a mature elastin contrast agent that also binds to tropoelastin. In the first clinical phase by now. We, as I said, in the group, in my previous group, there is a student working on a collagen 1 and collagen 3 contrast agents. We have also contrast, I mean, we have for so many things. We also have contrast agents that binds to specific receptors of macrophages, a bit of like every component that you think it might be abundant enough can be a potential target for MRI.
Okay, thanks. Nice, really nice. As I said, I have enjoyed a lot your presentation, but also your answer to these questions. I think that for me, it's enough. For the attendees, it seems so. So if you think, Maite, we could, you could give a last word if you like, and we can say goodbye. So.
Okay, I mean, yes, of course. I think that the only thing is thank you very much, Begoña, because it was a very nice talk, and I think that many questions are in the package. Then I think that maybe we can ask you later. But anyway, everybody has to know that next Thursday is going to be the next talk. I think that maybe it's going to be a very different talk because it's going to mix art and chemistry. Then I think that it's going to be very interesting talk. And well, and it's all. I mean, thank you very much for the attendance of everybody.